The overall goal of this experiment is to prepare high-quality enriched stomata guard cells for physiological, biochemical, and molecular assays such as transcriptomics, proteomics, and metabolomics. Hello, my name is Sheldon Lawrence. I am a graduate student in Dr. Chen's lab in the Department of Biology at the University of Florida. Today, I'm going to show you how to isolate guard cells from plant leaf tissue that are useful material for studying both the physiology and biology of stomata. This method is considered a novel method for isolating guard cells. It specifically allows for the enrichment of intact guard cells and provides reliable samples for the study of stomata cell signaling processes.
The application of this method can extend to Brassicanapas and other plant species with only small modification to the protocol.
The demonstration is important, and many steps to enriching the guard cells can prove challenging.
To begin, select mature 5-week-old Arabidopsis plants. Carefully remove a leaf with a scalpel, cutting the leaf at the base. Take 2 pieces of Scotch tape and place the excised leaf between them. Gently apply pressure to both sides while ensuring that the end of the tape pieces don't adhere. This can be done by inserting a finger between them.